We will start a small topic which is called insulin. Insulin is a hormone which has been synthesized and stored in the gland called pancreas. Pancreas is having two portions. One is exocrinic portion and the endocrinic portion. The exocrinic portion is nothing but the release of pancreatic juice directly into the GSNL tract, which is performing the duty of digestion of the absorbed food or the taken food. And thus the other part is called as exocrinic function, and not only the exocrinic function, it has the other function what is called endocrinic function. The endocrinic function having a habit of synthesizing and storing the hormone what we are dealing today is nothing but the insulin, and that insulin is being produced by a small part which is called acini, the last ductural part from the pancreatic ductules. And this acini is a combination of four cells which consists of islets of Langerhans, and these are all the other cells which consists of one cell is called as alpha cells, and these alpha cells release a hormone called glucagon, and this is just the reverse of insulin. And the beta cells which consists of islets or Langerhans, they are used for the preparation of insulin. Now insulin is one of the hormones which is always used for the cut down of glucose and diverting the glucose from the blood circulation to the various parts like tissues and other various organs. And the third one is called as delta cells, which is secreting a hormone called somatostatin, and this somatostatin is having a beautiful function. That means it is maintaining both the glucagon and the insulin for maintaining their purposes of their functions, whether to increase it or whether to decrease it, what is the function of glucose and what is the function of insulin against it, what is the function of glucagon and how it is the function of against the insulin. And the other one which is consists of is called as pancreatic polypeptide cells, and these pancreatic polypeptide cells are secreting a hormone called as pancreatic polypeptide, and these are also having a limited function in the functions of islets and Langerhans. Now we saw the four types of cells which are present on your acini part, which is doing the functions of endocrinic function. And daily if you see what is being synthesized and how it is being stored up, they are about 2 millions of islets of Langerhans in the acini. And when we see that for every 15 to 30 minutes, this insulin has been secreted, stored and synthesized, and that's stored into your particular vesicles of your acini, right? And if at all, if it is needed and has to be released into your blood circulation, it wants some stimuli from the body. There are different types of stimulation from the body. We can categorize it into three. One is called as chemical stimuli, other one is called hormonal stimuli, and other one is called mechanical stimuli. This chemical stimuli is nothing but the taking some of the chemicals which has been increased by its stimulation, and the hormonal function is nothing but the maintenance of glucose, how it is being increased. If at all your plasma concentration has been reached more than your 30-90 milligrams, as I mentioned over here, if at all your body concentration of glucose has reached a little higher of 90 milligrams, they are directly released and they are starting at that stimulation to release the insulin and store it in your blood plasma concentration. And later, if you have a lot of stress, that means if at all our body is under stress and is having some pressure on it, and it has to release one more neurotransmitter which is called adrenaline. And now whenever the adrenaline and the stress has been happened in our body, there is a lot of stress including in, and it needs a lot of glucose to be needed. So if you want to have the glucose needed, then particularly the stimulation of the insulin has to be inhibited so that the glucose is directly produced and give the functions of stress and the pressure what the body has been created to the particular situation. And so we can say that this has been regulated by, one is chemical, the one is hormonal, and the one is mechanical. And if you see that the process how it has been done, this stimulation, whenever our body is being stimulated by all this process and also by the glucose levels in the body, it is particularly stimulated from the blood circulation, taking it to the beta of islets Langerhans. Now I am zooming this particular beta cells of islet of Langerhans over here, where the stimulation has been reached from the receptors into the beta islets of Langerhans by our receptor calls, and these receptors are nothing but the insulin receptors. And these insulin receptors are not only doing the functions of two cycles perfectly, what is the Krebs cycle and the glycolysis cycle. And we know that the end products of glycolysis cycle is nothing but having lots of ATP molecules, production of ATP molecules. And now these ATP molecules has been developed inside it, and they are diverting themselves into RER. What is RER? RER is nothing but your rough endoplasmic reticulum. And this rough endoplasmic reticulum is storing or trying to stimulate the production of insulin in the form of pre-pro insulin. We know that the insulin is in the form of precursors, and it is not the direct product, it is a precursor. So the first product of insulin is called as pre-pro insulin, which is being done it in your RER, nothing but your rough endoplasmic reticulum. And it is also a product of 109 amino acids. And from the pre-pro insulin, they are directing themselves to the Golgi apparatus of the cells, where it is being developed into called as pro-insulin, which is having a characters of 86. And now this 86, which is called as pro-insulin, has been secreted in the Golgi parts. And this Golgi apparatus are being collecting all the particles and develop themselves into pro-insulin. It has been stimulated and once again taken by the cells and enters into the mitochondria, and where this pro-insulin is directly converted into called as pro-hormone or the insulin hormone, and this is taken up by the mitochondria, and a lot of ATP molecules are produced over there. And once these ATP molecules are produced up, they are entering into your cell part, which is being secreting your secondary messengers, and the other processes already we know that. And immediately the insulin is started synthesizing and poured it up into your beta flagellantes and it's directly storage over here. And depends upon the stimulation released by the body, whether related by your chemical, hormonal, mechanical, as we said before, they have been do it into two portions. One is called as rapid portion, other one is called as delayed portion. In the rapid portion, that means the word is showing you that there is lot of speed that has to be released over there, so that it takes up all the glucose what is being needed by the body and divert themselves to all the tissues. So immediately it takes up lot of insulin stimulation and directly release the insulin part, whichever is being released out, and directly poured into the circulation, which maintains your glucose level. And the other one is called as delayed portion, where it is a delayed means the name is showing you that a small slow process has been done. Stimulation is being done it in a slow process. Here two portions has to be indicated. One is for the creation of new synthesis of insulin, and the other one is the release of stored insulin already which is present in your beta glucose. So this is as two processes faced by the delayed portion. Now we up to now we understood that how, why and why the insulin has to be secreted, how it is being stored up, and how it is being made up into the blood circulation with the two processes and all. Now we'll see what is a insulin. And insulin, if you see, it is called as a amino acid accumulation product, which is having a protein in nature and divided into two portions. One is your A chain and the other one is called your B chain, that is included over here. This A chain consists of 21 amino acids and the other one B chain, it consists of 30 amino acids, and both these are combinations which is starting with the A and the other one which is starting with the B, and these two are being connected or cross-linked by another chain what is called C chain, and this C chain is being the link is made up by bonds called as disulfide bonds, and these disulfide bonds keep both the A chain and the B chain keep it into the situation, and whenever the insulin has to be break up or whenever the insulin has to be degenerated itself, this bond has to be cut, and this cutting of C portion of chain has been done by a enzyme called as insulin protease, and when this insulin protease is helping, this chain has been cut and it is being released into the blood circulation to perform its own functions, that is the regulations of your glucose. Here one more point has to be remembered. What is that? Not only your insulin is controlling your glucose part, that is your carbohydrate part, and not only it is diverting or increasing its reuptake, diverting to the tissues part, it is also maintaining a protein part or the protein diversions. How it is? It is using to decrease your glycogen breakdown content where the protein has been involved, and later it is also doing the action of free fatty acids. That means it is trying to inhibit the release of free fatty acids from the fat cells so that the glucose cannot be created inside your blood circulation, but still it is stored into the particular bodies. And the free fat cells, we know that they are adipose tissues. And here we can see the glucose conversions also. And we can see here the functions of insulin. And this insulin is doing, the first function is decreasing of blood sugar level. That means it is diverting that blood sugar's level reuptake by the different tissues. That means it is diverting its functional. That means each and every tissue wants some energy, isn't it? So if at all the glucose has been released and diverting it to the tissues can perform its mainly the physiological functions what they do. And they are also been increasing the storage of glucose as glycogen. Here we can understand that the glycogen is a storage part where the glucose is converting into glycogen. This is called glyconeogenesis. And it is also decreasing your glycogen breakdown content, and it is increasing your amino acids reuptake. That means it is taking the reuptake up by the muscles so that the muscle tries to function more the functions of both the contraction and the relaxation. It is increasing your protein anabolism, and it is maintaining all the anabolism of proteins, nothing but the creation of glycogen. And the glucagon, which has been secreted by the alpha cells, is also maintaining the functions of glycogen also. So it is doing two functions. One is it is decreasing your gluconeogenesis and it is increasing your glycogeogenesis. The glycogenesis is nothing but the conversion of glucose as glycogen, and the decreasing of gluconeogenesis is nothing but the diversion of all the proteins, carbohydrates and fats in diverting itself into lessing parts. That means it is trying to cut down all the process of so that the glucose can be transmitted to the blood circulation to increase the power. Why? Because it is almost increasing your blood circulation. Now we'll see what happens over here. The functions of insulin has we just discussed now, and it is the doses which are available over here are about 40 units and 80 units. And here always the insulin are measured in units. And so this is just the introduction of insulin, and just later in the next classes we'll try to see how the diversion is being done to the adipose tissue, how the diversion is done by your fatty cells, how diversion is done by your glycogen, and how the diversion has been done in the carbohydrate content can be seen with a beautiful picture in the next class.